I'm Dr. David Maloney from the Hutchinson Cancer Center in Seattle, Washington. I'm joined by my colleagues here from the first day of the lymphoma session, leukemia session at iwCAR-T, Dr. Noelle Frey from UPenn and Dr. John Gribben from Barts in London. And I think we had another really exciting day one. We started it off with, I guess, where CAR T cells really kind of started off with ALL. I mean, what do you think the highlights of the day were?
I think the ALL session was really inspiring because, just as you said, is kind of where it all started. And all of our discussions today kind of acknowledge these initial successes that we've seen with CAR-19 and ALL. And it was really about the next steps, how we can make things better. And so, we talked about using humanized CARs, dual targeted approaches, off-the-shelf CARs, and had a lot of discussions about different approaches to optimize all those potential ways of improving relapse-free survival. So it was an inspiring session.
Yeah, I was struck by what we've learned in the relapses, right? I mean, this whole concept of being able to tease out the patients who have sustained CAR T cell activity with consistent B cell depletion, and then they're the ones that are relapsing with loss of antigen, whereas the other group that's relapsing antigen positive, the problem is probably the T cells, and so those strategies really, detail different ways we could approach it. I thought that was fascinating.
I completely agree, and I think what was clear is that we're not going to have one solution because the problem is very different. So there's not going to be one solution fits all of those. But then what we really need, isn't it, is a way of identifying quite early on exactly what the mechanism of failure is to be able to come up with a strategy that addresses why that particular patient failed. I completely agree with you. I thought coming into the session, gosh, what are we going to learn new about ALL when we've had one of these every year now? This is their fourth year, and we've had one every year. But every year, there's even in the disease where we think we know the most about it, there's more stuff that comes out.
Yeah, and I think it just highlights how, we're just never, for our patients, we're never satisfied with things are so much better now than they were 5 years ago. We're 4 years into it and we're making all these advances, and it's exciting to think about, where we will be, in a few more years with ALL.
Yeah, that actually led to a lot of interesting discussions at the end of the day where we were trying to figure out where will we be in 2027, for example. And I think there's a lot of speculation of how to move CAR T cells earlier, especially as you were trying to do in ALL. And I think the other approach is, like, how are we going to get combinations approved, or the multi-targeting CARs, or the human CAR? I mean, these approval strategies are challenging. Now to turn to something that was actually a little more challenging, AML. We're still waiting. What did you think about our progress in AML?
Yeah, I mean, it's really difficult, thinking both about the CAR-Ts and the bispecifics. What we're hearing is, of course, we don't have a CD19 equivalent in AML or anything near it. And if there's heterogeneity in terms of complexity of what should be the target in AML, we're not even established what the target should even be in AML yet to know what we're doing. Also, the whole approach of what it is that we're trying to achieve with immunotherapy. Are we talking about it exclusively as a bridge to transplant? Or are we potentially looking to develop this as a curative strategy? I was hearing most of the studies today about how do we get a refractory patient as a bridge to a transplant. So I think we've got a long way to go. And of course, almost all of that relates to how similar the AML blast cell is to the normal hematopoietic. So where's the therapeutic window for immunotherapy to be able to come in and target those cells?
We just don't have good antigens that can distinguish between the two. So that led to, again, interesting discussions about whether we should just ablate hematopoiesis and then send people for transplant. But seems like there's some progress being made, but I don't know. We've been waiting quite a while for AML. We're actually starting some trials now, so I think it'll be interesting.
Well, I mean, it's some really neat approaches. What SAR's doing at your place. Knocking out the CD33 to bringing back hematopoiesis that doesn't express the target. But even SAR said openly, maybe CD33 is not the answer, but there's at least a creative step towards getting to what we want to do, which is at least to replace the hematopoiesis, which enables you to at least go in and continually target this without the toxicity that we see associated with targeting something that is expressed on normal hematopoietic progenitors.
And there's just such an unmet need in our AML patients, and I think we saw a lot of kind of redefining the goals, like you said, to get patients to a better place. And I thought it was interesting in part of the discussions, people mentioned that it might be, if our goal is to myeloablate and get someone to that transplant, it might be an interesting role for an off-the-shelf CAR, because you don't have that goal of long-term persistence, because it's getting you to your endpoint of remission, just because it might not be feasible.
So the other major disease we talked about, well, your favorite disease, I guess, is CLL, and it sounds like there's some hope on the horizon potentially with CARs.
Yeah, finally there's some hope on the horizon. You said, of course, we started with ALL, but of course we started with CLL. Carol June and David Porter's first report out of UPenn was actually the ALL, and of course such hope that those first two patients did so well. Unfortunately, we've had some major setbacks along the way. I think all of us believe a lot of that has been to do with the T-cell dysfunction that's so intrinsic in CLL. And of course, the other reason it's probably getting a bit better now is that the chemotherapy that we're using for CLL, the fludarabine-based regimen, nothing's more immunosuppressive than that. So trying to collect fit T cells from CLL patients has been a real challenge. And of course, what we spent a lot of time, maybe too much time talking about today, because we ended up hearing 3 different approaches all talking about the same thing, which is how a BTK inhibitor, namely ibrutinib, is able to very clearly improve the fitness of the T cells that you collect. The issue, I think, that we've got in CLL, as in pretty much every other disease, is once we get the CAR T cell approved, who and when in the patient's journey is the right approach. If you wait too late, we've all seen when patients have failed these therapies, the disease can really accelerate very quickly. When patients develop mutations at the BTK binding site, those patients can really accelerate. The risk of Richter's transformation and how you can't salvage those patients back mean that you can miss the window of opportunity that a CAR T has. But with the successes we've got, with the current treatments we've got, offering a CAR T too early also raises its own ethical issues. So, but what we did hear from the companies today was, a very clear timeline of when we can expect at least Lysocell to be commercially available, so at least we'll have a product that we can, as we've done in other diseases, start to explore when and in which patient population we're able to do this outside the setting of clinical trial where that's defined for us. And I think as in academic centres, I think we're very well equipped to answer those types of questions.
Yeah, I was struck by the fact that, most of our patients have still been treated with multiple regimens, and then the UPenn experience recently with ibrutinib, in fact, some patients were frontline patients with only 6 months of ibrutinib and went directly into CAR T, and there at least the survival was 100% in that population. So, I think that does, if we can get a CAR T cell approved in the space which we desperately need, then we can start trying to see whether can we eradicate MRD. Can we do that with no toxicity or with less toxicity? And I think that'd be a great advance. I was kind of surprised about the discussion with ibrutinib, in that I kind of thought, maybe naively, that this was clearly a TKI effect on T cells. I'm now not so convinced, because you could equally say it's the TKI effect on the CLL cells modulating their impact on the T cells. And that's an area I know you know a lot about, but do you think it's more just the cytoreduction of the disease or the change in that histology, or do you actually think we're modifying T cells? I think it's
probably almost certainly both, but there's a huge amount of data out there now. So ibrutinib and all the BTK inhibitors down-modulate expression of PD-1 on the and a PD-L1 pathway. So there's very indirects it can have. BTK is highly expressed on myeloid cells, and particularly on myeloid derived suppressor cells. So there's a whole complex way in which these agents can modulate the whole microenvironment, which of course is where, if you get those CAR-T cells in there. So I think it's much more complicated than just the fitness of the T cells alone. I think it does impact those T cells, but I think it impacts the whole tumor microenvironment and induces a much more, better environment for those T cells to be able to have their activity.
And I loved how one of the things that came up in the discussion was using what we've learned about the CAR-19 for CLL with ibrutinib to potentially apply ibrutinib to other diseases where ibrutinib isn't not necessarily a standard of care to treat the disease. And I think just highlights how helpful this type of meeting is, because we're kind of hearing things from other subspecialists.
I think that there was a study done at Penn, well, skip through Penn, Steve Schuster did a study with ibrutinib in large cell lymphoma. And
well, at least you can make a rationale, because of course there's been studies done of thinking that ibrutinib might have a role in, certainly, from the work that Lou Stout's done, at least in ABC subtypes of diffuse large B-cell lymphoma, which are highly likely to be the ones that land up going to CAR-T. But what we're talking about here is diseases in which there's no expression of BTK. There's still the potential to be using those agents to modulate the immune environment. You can imagine doing this in myeloma and in ALL and in solid tumors. And I think there's every rationale to believe that those would be effective.
So I think we saw a great day on those three diseases, and then we kind of finished it up with some wild speculation. What were your thoughts from that effort?
Well, I think both you and I were a little bit worried about, this was an idea we had of just, let's throw some ideas out there about where we're going to be in 5 years, and you and I thought this session could be over in 5 minutes. But in fact, right from the very first question stimulated some great discussion among us and among the audience. I still wish our colleagues from the pharma companies would get up and be more involved, because lots of those questions would have actually been better answered by them, and I wish they just would stop being quite so shy and really kind of step up, with one notable exception. But
But interesting, I did get feedback about, wow, this is such an open discussion. You guys are just like, because there's no holds barred there in terms of what people are asking. Not coming from pharmacists.
Well, that's the whole point. This is why it's a workshop, and this is why we don't want this meeting to be 500 people. The concept of being able to talk openly, have the time to discuss it, and have, as you say, what I love about it is we're so used to going to a meeting in our own disease, and just hearing how other people are thinking about the same problem from a slightly different perspective just opens your eyes to different opportunities, doesn't it?
And this was pretty informative, because yesterday we had done the Solid Cancer Day, and a lot of those people stuck around, and there was a lot of cross-fertilization. So I thought this was a really outstanding meeting to get back together. It was partly hybrid. We did have probably 20% of the talks virtually, but that actually worked quite well.
We also had a big audience who were just watching virtually.
But in terms of the speakers.
Oh, in terms of the speakers. But I'm always worried about, I think personally that hybrid meetings are the way we're going to go forward. I don't think we're ever going to come back to the day where everyone's going to get on a plane and travel halfway around the world to give talks. And I think as the technology continues to improve, I think it worked pretty smoothly today, the transition between those speakers who were presenting virtually. In fact, you could argue that they're the ones whose talks were usually the 10 minutes. But even in terms of the interactions we were able to have with we had a couple of issues with the ALL side, but once we'd overcome that, the technology's there for people to be able to have a fully interactive process. And I'm the one who's kind of stressed the importance of doing a workshop. I didn't feel that the people who were there virtually were not contributing towards the discussion.
Yeah, I agree. I think going forward, we're going to be able to have these workshops with this hybrid method just be more inclusive and more interactive, because COVID has pushed us all to make
it's the gift that keeps on giving.
Engagement of people who aren't on site, meaningful and productive. So I thought it worked really well too, the hybrid model.
All right, well, thank you both, and we'll look forward for day 2.